technique enables a reproducible and statistically valid determination of starch granule sizes and provides a proper description of statistically distributed granule sizes. This technique determines starch granule volumes independent of their shapes and uses a statistically valid granule sample sizes for size distribution and provide an improved specification of granule size distributions.
This method is applicable to plant and food science research studies that involve starch granule sizes, in addition to any other starch applications that require granule size information.
Before beginning an analysis, dissolve 25 grams of lithium chloride in 500 milliliters of methanol, and select an aperture tube with a particle diameter range covering the known granule size range of the starch samples to be analyzed. To set up a standard operating method, start the analyzer software and in the main menu, click SOP and Create SOM Wizard and select the appropriate settings. Then click Edit the SOM and select the appropriate settings. To select a preferences file, under SOP, click Create Preferences Wizard and select the appropriate preference settings. Then click Create SOP Wizard, enter a description and author, and select the SOM and preferences files to create and save the SOP.